So the PAS-1 trial, PAS stands for pancreatic adenocarcinoma signature stratification for treatment. And what we're trying to do is try to understand, are there any genomic, RNA, or other biomarkers that we can pull out very early in the diagnosis of metastatic pancreas cancer and use it to inform treatment for these patients after the standard of care chemotherapy. And maybe even more importantly, learn from that who responds better to what type of chemotherapy so that we can improve the selection up front for patients going forward. And so that's the trial we're presenting. So it's interesting, when we started the trial, we made the assumption that while both chemotherapies that we have today, FOLFIRINOX and gemcitabine nab-paclitaxel, are very useful, the assumption was that FOLFIRINOX would be a little better. And so we powered it, assuming that we would see a slightly longer signal for FOLFIRINOX than the gem nab-paclitaxel, but we would learn how to use each one of them perhaps better. What is a little surprising is that that's not what we showed. We showed that there is essentially very little difference between either of those chemotherapies. And in fact, the gemcitabine nab-paclitaxel came out as a little bit ahead, a little bit longer time for the patients to progress, and a little less toxicity. Now, having said that, I don't really think either of them are great. They both leave much for improvement. But it opens the discussion that we shouldn't be hanging our hat on one or the other. We really should be trying to choose from the beginning what will be best for this patient and then building on that. Well, it'll be interesting to see how this impacts clinic. I hope this gives oncologists a little bit more comfort knowing that it isn't wrong to choose one or the other. And if there is a trial that is using one of these for good scientific reason, then that makes sense. But to avoid one or the other without a scientific rationale doesn't make any sense. What we're hoping is that as this data matures a little bit, we'll be able to strongly say, you know, you can pick certain subtypes of pancreas cancer right from the beginning. There's something called the classical, there's something called the basal, and this you can tell in the RNA signatures. And right from the beginning, you know, this one will probably be doing better on gem nab-paclitaxel. Let's add a KRAS inhibitor. Let's do something innovative on top of that rather than just waiting for a subsequent line. One of the other findings in the PAS trial is, although we've done all this intensive science on every patient, the reality is that only just over 50–60% ever get a chance to even use that and move to second line because they deteriorate so quickly. And so moving all that information up front and using it earlier on is likely to impact patients a lot more. I think we're moving into a very interesting time. I don't mean to say that I don't think chemotherapy has had its role. It's been very helpful, especially these combinations, but we have to use them smarter. We have to know who's going to respond to one and not to the other, and then we have to start adding, you know, novel targeted agents, KRAS inhibitors, different things like that to the chemotherapy and optimizing each patient. So we're not getting 60% getting to second line, we're getting 100% to second line, and then they're doing well as well. So we're moving into good times, I think, for pancreas cancer, and I think the past data will be very informative to try to build those next steps.